I think maybe we'll just have to decide, do we want to move forward or not, and take that bill off the wall, have Ledge Council actually draft some language. So be thinking about that as we hear this testimony and tomorrow's testimony, I guess.
In short form?
Yeah,
Oh, okay.
yeah. So with that, John, I know you said you might want to kick things off.
Just briefly, my role here is pretty nice.
Okay, all right. Well, why don't we have you come on up then, and we can, yeah, feel free to take that chair.
There we go.
We can go ahead and get started, and we'll do that by introducing ourselves, because it's been a while, at least since we talked, and I'm not sure that you've been in this year.
Last year.
Last year, okay, yeah. Well, we got some new faces. So I'm David Durfee. I live in Shaftesbury in Bennington County, and I represent Sunderland and Glastonbury as well. Go around this way.
I'm Leland Morgan. I represent Grand Isle County, the whole county, and the western portion of the town of Milton, Ohio.
I'm Jed Lipsky. I represent Lamoille 1 district, live in Stowe, represent Stowe.
Michelle Boslin. I live in Westminster and also represent Rockingham and Brookline in Northern Windham County.
John O'Brien, I represent Royal County.
Heather Supna, I represent Barnard, Pomfret, Bridgewater, West Hartford.
And Sidonia is our committee assistant. If you have any material that you haven't already sent her, you can do that. I think we do have this presentation is on our website, on the committee website. We've got two committee members who are not here. One, Representative Byrd is out today. Representative Nelson had to step out. I don't know whether he'll be back or not, but let's turn it over to you then.
Well, good afternoon, everybody. My name is John Austin. I'm the director of the Wildlife Division with the Vermont Fish and Wildlife Department. Pleasure to be with all of you today. Bree Furphy is our fur bearer project leader. She's one of our wildlife scientists in the department, responsible for all things fur bearer related, everything from coyotes and foxes to fisher and bobcat, and you name it. Bree has a lot of experience dealing with these sorts of animals and is knowledgeable about the topic that you've taken up, that is the presence of rodenticide compounds in wildlife. She's going to focus today on mammals rather than birds, but as I think you've probably already heard from other people who have testified, that this is also an issue for birds in the avian community. I guess I would say, just as a general statement on behalf of the department with respect to the bill at hand, we share the committee's interests and concern regarding the presence of these substances in the environment. We work closely with our colleagues in the Vermont Agency of Agriculture to deal with issues like this. We intend to continue to do so on this particular topic moving forward. I think the overall goal is to come up with strategies that are effective at minimizing risk of exposure of these substances to wildlife. And I think there's some work to be done in that regard, and that's what Ms. Furphy will share with you today. So, Bree, I'll turn it over to you. You're the expert. You can share your deep knowledge with the committee, and then we're available for questions.
Great, thank you.
Thanks for having us.
Okay, thank you for having us. My name is Bree Furphy, for the record. You guys already learned about me a little bit. I'm the fur bearer project leader for the Vermont Department of Fish and Wildlife. So I would like to give just an overview of what we've learned about rodenticides here in Vermont with some of our mammal species that we have. And then in addition, talk about what we don't know and why we don't know it. And then talk about a little bit what we're doing now and some of our plans for the future. It's a really complicated subject and it's not quick, cut and dry. As you'll learn, it's very nuanced and there's still a lot more to learn. So as you all have probably learned, anticoagulant rodenticides, they interfere with the vitamin K mediated synthesis of blood clotting factors in the liver. I believe you all heard testimony about what that is like. So if an animal had gotten anticoagulant poisoning, a lot of times you can get vitamin K to reverse the symptoms. There are studies that show that ARs or anticoagulant rodenticides may suppress reproductive capacity or immune function, may increase vulnerability to morbidity factors such as parasites, disease, and predators. One thing that's been documented is the prevalence of mange with exposure to rodenticides. And not only is it uncomfortable for the animal that has mange, but mange in severe cases can lead to mortality. So with other pressures that are going on in wildlife populations, such as global climate change and issues with habitat loss, this could be a really important challenge for wildlife going into the future. And importantly, lethal concentrations, they can vary among species and among individuals. So it's really difficult to ascertain what are lethal levels of one thing because different species and individuals are affected differently.
Before you go on, Bri, can you just talk a little bit about the top bullet point, blood clotting? I mean, I think we understand that anticoagulant means that blood won't clot. Does that matter when there's no wound?
Does that matter if there's no wounds? Yeah, it does matter.
Can you just speak to that a little bit?
Sure. So for example, if you are looking at an animal that you're doing a necropsy, kind of like an autopsy on, if you open up the animal and you see blood in the body cavity, that would be a really good indicator that perhaps there's AR poisoning because the blood has not clotted. So you would see that in animals that, regardless of them having injuries, they would potentially bleed more, but it depends on, of course, the level, the severity of the type of poisoning and it's the compound itself. Can't say for sure because, like I said, this is really complicated and there's so many different compounds and different ways that it affects wildlife.
So they just internally bleed without any outside, just.
Yeah.
Is it always in the liver where the bleeding takes place, or do you see any other organs in the center?
So.
Seeping or.
So I wish I had a photo. I just recently took a photo, but the liver is actually the best sample that we could possibly get because that is where all of the compounds bioaccumulate over time. And so by taking a liver sample, you can get the whole history of exposure compared to something like a hair sample or blood samples or even fecal samples. That will not give you the complete picture because that will only give you the recent exposure. So the liver is actually the best way to be testing the complete picture. When you're looking at, when you're looking at an animal and you're doing a necropsy, you could tell some of the hemorrhaging that occurs in the organs. And so you might look at different health indices such as, okay, what does the liver look like? What does the heart look like? What does the lungs look like, for example? And if you're seeing some of that hemorrhaging that's unrelated to something like a trauma, like a car crash or, something like that, then it would be indicative that there could be something else going on.
Representative Nelson?
Yeah, sorry for being late. How many animals do you receive a year that you trace back to vitamin K? I'm sure you probably went over it with the committee.
So, well, here we go. So kind of transitioning to this, typically every single year we receive approximately 100 to 150 fisher.
Yeah, from trappers turning in.
From regulated trapping.
Yeah.
Mhm. So there is a regulation or a rule in place where it's mandatory to turn in fisher, otter, and bobcat carcasses to the department. Through those carcasses, we're able to conduct a variety of different studies, the necropsies that I was just speaking about, and test for contaminants. So we studied 3 years in advance, 3 years prior, so from 2018 to 2022, so 3 trapping seasons worth of data. We sampled 71 Fisher and 29 Bobcat from those 3 seasons. And every single one of those Fisher had exposure to at least 1 or more rodenticide compounds. And Bobcat, the majority of them had rodenticide compounds, but not every single one.
Okay, and do you report these to the Agency of Agriculture?
Yes, we do.
You do.
Mhm.
Representative Bosland.
Yeah, I guess I'm just curious if, are people intentionally trapping Fisher and Bobcat? These are accidental trappings? Like, if somebody's trapping, why would they then turn it into you? Like, what's the point in,
So,
are they considered pests? Like, I mean, how does I don't understand why somebody would kill a Fisher and then give it to you. Like,
Sure, yeah, I'd be happy to
Okay.
explain that.
Yep.
So for regulated trapping season, we have Fisher, Bobcat, and Otter seasons. And so they're not considered pests. This is not from people that are trapping for nuisance reasons. Typically, those 3 species are generally not trapped for nuisance reasons. That's more like skunks and raccoons. But it is a rule to turn in any carcasses of those 3 species to the department.
and then
For
do you give them back?
But not the hide. No.
not the hide. So yes, we do not get them back because we conduct full necropsies. And what we do with that information is that we collect age and sex data. And so it helps us get a complete picture over time of what's the age and sex distribution of the population. But the benefit of these carcasses is that it's free to us to sample. And there's many universities over the course of the last 3 decades that have collaborated on a variety of different studies from these carcasses.
So what's the motivation on the part of the trapper though? Are they going to be making something out of the hides or what?
Yeah. I mean, there's a variety of different motivations for trappers, but they could be trapping for monetary benefit. They could be making products from their hides. You could eat bobcat. I hear that it's very good. I've had cougar before and it's strangely enough, very good. So
It is.
somebody might be capturing for food reasons. But there's a variety of different motivations for why somebody would want to capture one of those 3 species.
Just one last question. What kind of traps are they using for these for the most part?
Depending on the species, you can use body gripping traps or you can use body gripping traps. So there's different types of traps that basically are more appropriate to use for some species more than others, like otter, for example. You would want to use a body gripping trap in that scenario because it would be the most efficient way to capture an otter versus something like a foothold trap or leg hold trap that basically the animal would walk into. So the trappers would want to use the most efficient and humane method possible for using traps to capture the animal.
Representative O'Brien.
What happens when something like a Fisher eats a mink or, maybe a predatory raptor, say, that has concentration levels? Is it any different than eating a rat that has concentration levels that they are?
Well, it's a good question. And it's hard to compare, right? So of the Fisher that we've tested thus far, all of them came back with exposure to at least 1 compound, right? Or traces of compounds. We haven't had any to really compare that haven't been exposed. So the impacts of eating the rodents or other species as prey items that have been exposed to any of these compounds, I mean, it's going to be pretty variable. It could have no effect or it could be lethal. We don't know. It depends on the compounds that's being ingested. It depends on the health of the animal. It depends on the age and the sex of the animal. And Fisher is just one species. It could be that, foxes and coyotes are also being impacted.
Representative Nelson.
You testing Fisher that just caught on the edge of town? Because you go back in Essex County. I don't think there's many people using these rat baits. I could be wrong. Maybe people are throwing them around their camp illegally and not monitoring them. I find it real hard to understand that a fisher that runs up on the Lowell Mountains is gonna be contaminated with this kind of thing. The range is big, but it's not that big. I mean, we don't ever see fisher down on the farm. Don't ever see bobcat on the farm. Certainly, if a coyote's on the farm, it becomes endangered. But I'm just saying, I don't know. I can't understand why your number's such a high percentage have these kind of compounds in them. And I just wonder where your sample's coming from, if your sample's broad enough to, if you're really looking to see if there's an issue, you should almost check every liver from every fisher. And how many you get, are they turning in a year now?
So approximately 100 to 150.
Yeah, so about running pretty steady.
Mhm.
Yeah,
So your sample then is somewhere around maybe a quarter of the total,
Not even.
not even.
Not even, maybe like 1 to 2% at best. So, to speak on Representative Nelson's, question, we do, when people turn in the carcasses, they do give town information that they
Mhm.
harvested. So we do have information in terms of what towns have higher exposure levels, what towns are having more exposure levels for different compounds. That information is available to us.
yeah, it'd just be interesting to see, because I realize you're testing about 25%.
Maybe a lot less.
Well, 30 out of 125, 125 to 150 animals, but 11 in the last year. And I know there's a funding thing too. It costs money to run these
So.
tests.
Yeah, so 71 samples over the 3 years, right? So each year, we're seeing that there are multiple fish are being exposed to more than just one compound, which is why you're seeing numbers that are larger than 30 there. So that's pretty significant because it basically insinuates that the fish are being exposed at multiple times. And it could be that it's happening in areas that have homes. We don't know. But there's a lot to figure out there for sure.
Yeah, of course, I know how fish are trapped. I'll explain it to Michelle off to the side later, but I understand, I have trapped. And, you can't do it from just looking at the carcass. And I'm glad you're doing a liver biopsy. Okay, yeah.
Representative O'Brien.
Are some of these compounds passed through, say, we're talking about mammals, mother's milk or blood in the uterus so that the young end up with it?
I do not know. It's very, how would we know? It's very difficult. So I wanted to point out though too about these different compounds. So Broda is actually a second generation rodenticide. So those are the ones that are more potent and typically would bioaccumulate pretty fast, generally in their prey species. And then diapacinone, hopefully I'm pronouncing these correctly, that's a first generation. So that alone is fairly interesting because it's like, why are those two standing out more than others?
Decon.
What's that?
Decon. First generation. Everybody used Decon. They had that little cardboard box, they flipped it open and they set it everywhere.
Are you suggesting, Representative Nelson, that Decon contains one of those
Yeah.
highlighted?
First generation. I could look it up, but I wouldn't be surprised if that's what it was. And of course, it'd be, you had only 11. Of course, that's an old number that the agency has since doubled down on how these things are used. Very much so.
for clarification, the carcasses?
Yeah.
Or, oh, yes.
Yeah.
So, I will be getting to that, but yes, we are
Yeah.
continuously monitoring the
Okay.
situation.
Yeah.
Representative Morgan.
Do these compounds break down over a period of time? So if an animal had a certain amount in him and he got no more intake, does that break down over a period of time and pass from the body?
My understanding is that it's definitely a possibility, but again, it depends on the compounds and the health of the individual and what's going on. So, like, for example, if we send samples out and there's traces of something, it could be that there's much higher levels at the time of ingestion for a period of time that we just don't know about. So it's really difficult to say with any sort of certainty.
And Representative Lipsky had a question.
And I'm going back a little bit. You said that and maybe I got it wrong, I didn't understand, but you only found concentrations of these rodenticides in 1 or 2% of the animals
Of the population.
of the population.
So the harvest that we typically have from year to year is very minuscule compared to the entire population that we have across the state. And the samples that we are getting are well distributed. So during this study, there were target areas to sample from to make sure that we were sampling from representative areas across the state.
Geographic.
Mhm. Yeah, so this isn't just coming from, say, like southern Vermont. This is well across Vermont.
Let's take one more question and then we'll let you
Follow
go
up.
on. Yeah.
Just how many fishers say, or martins, because we were just talking about martins, are in the state?
Well, it's hard to say. We do know that they're very abundant, because we have cameras all over the place and they are probably number one that show up on our cameras. Yeah, they're incredibly abundant. We were just talking about martin. We just finished doing a study and trying to get martin on camera and all we got were fisher. And yeah, so, they're pretty widespread and abundant throughout the state. But we also have other indicators, and metrics that indicate that they're doing well in Vermont. We're not seeing any sort of population declines or any sort of big fluctuations that would indicate any sort of issues. In other states, that's not so much the case. But here in Vermont, we're doing okay.
Is this martin or fisher?
Sorry, that's fisher.
Fisher.
Yeah, martin are an state endangered species.
If fish are weighed 100 pounds, we'd all be dead. They are slick.
We would, yeah,
Yeah, we would be. They are slick. They move through the woods. They don't make a noise. They are slick.
How about the population of bobcat? Do you characterize that as healthy?
Yeah, absolutely. It's very similar. There's some areas that are gonna have denser population numbers than others. Addison County and the Champlain Valley have more abundant bobcat than other places. But generally speaking, the population is doing, from what the data that we have, that the population is doing really well.
And are they at the top of the food chain of what are their predators? Bobcat and fisher.
So, yeah, so bobcat are definitely amongst the top of the food chain. And fisher are amongst some of the top of the food chain too. They will share resources. How they compete for those resources really just depends on how they're using the habitat and what season it is. But generally speaking, like fisher, they can be generalists. So they can eat a variety of different food resources, whereas bobcat generally are carnivores and are going to be focused more on prey items. Fisher will eat carcasses, they'll eat mast material, so they, bird nests, things like that. Whereas bobcat are a little bit more focused on what their prey items are.
Turkeys.
Oh, that's right, turkeys, yeah.
Yeah, bobcats are cats and they're obligate carnivores.
We have a lot of them in there where I live. Bobcats, I see them very often.
I found a deer once killed by a bobcat and confirmed.
Was there anything else you wanted to say about this slide?
No, I just wanted to point out the two compounds that are being detected the most during that time period of sampling. Each lab, so when we send out the samples, they test for certain compounds. And so this is the complete picture of the different compounds that were tested for at the time. So the Department of Fish and Wildlife, we have worked pretty hard on sharing that information with other states, and the goal being to better understand this with Maine, New Hampshire, and New York. And so we put into what we conducted several studies, two that were published recently regarding the results that I just showed you and the other states, and what exposure levels are like and what could be driving those exposure levels. This was a graduate student project that was done exceptionally well and shed a lot of really interesting information that we wouldn't know otherwise. So those trapper-derived carcasses, all the samples that were done during these studies were from trapper-derived carcasses. And so without those samples, these studies weren't able to be done. We wouldn't know what we know today. I'd say also that in these publications, it's clear that the where the majority of the exposure is occurring is the wildland urban interface. And so that's defined as the houses that are, in more rural areas. That's where exposure levels were the highest. So that gives us a starting point, right? So we have that information. We know what this looks like comparative over the Northeast. We know how Vermont compares, right? And we know where exposure could be occurring the most. So in this public, or in one of the publications, I thought this was really interesting to include, but the left graph here shows the different compounds and the number of detections that there were across all the different samples that were tested across the Northeast. So the gray is actually the that it was detected trace amounts, and then the black bars are the amounts that actually had levels that were more detectable. So it's usually measured in parts per million, so ppms, and it goes to show that several compounds are detectable, traces of them, but then there's some that are showing up that have higher concentrations. The graph on the right there shows the differences in the state of harvest. Vermont's there in the middle. And in the red so the red bars or the brown bars there, those are all first generation. The blue shades are all second generation. So based on the results that I showed you earlier, you could see those two compounds showing up higher than other compounds. And it's very similar to other states. They're seeing the exact same thing, except for Maine. And in the publication as well, there is some predictive modeling looking at the exposure rates and also the number of compounds that Fisher would be exposed to. So Vermont, really, southern Vermont looks like it's a bullseye there, but it's looking like there's a huge section even beyond Vermont that could be potentially have issues going forward. We don't know. This is predictive modeling, and it's showing that there is a very high risk of having more than just one compound being detected in Fisher.
The southern New England states were not part of the study.
So we had New York and Connecticut. That was part of the study as well.
Connecticut isn't showing up any color.
It's, I
Oh,
think
I mean,
the
I'm
harvest is not, yeah. Connecticut, unfortunately, is experiencing declines in their Fisher population, and so is New Hampshire. And so if there's a decline in the population, you have, you could change the regulations to help the population.
And, the yellow in Pennsylvania is in the Allegheny National Forest. And the northern, unincorporated portions of the northern Maine. Is that because there weren't many tested or harvested there, or?
It's not that there wasn't that many harvested there. It appears that it has more to do with the wildland urban interface
Right.
that was modeled.
More.
Yeah, because the black dots represent the samples.
Do fishers eat dead animals too?
Yes.
It's in there.
Oh, yeah, they love it, especially during the winter.
I was wondering about something like the porcupines, for example. So like if somebody poisoned a porcupine, they might eat that too.
And you thought of that.
So one of the pieces of information that I think is really important to this entire conversation is that there's so many unknowns. Unfortunately, a lot of the questions that you have are the same questions that we have. And this is due to a variety of reasons. Again, different compounds affect different species in a variety of different ways. Individuals might be affected differently, age, sex, location, habitat quality. I mean, there's so many different variables that could be impacting wildlife beyond just the exposure itself. So that is pretty crucial to understand if we're trying to get at how are they being impacted. Because you can't actually take Fisher from the wild and put it into a laboratory setting, right? We can't figure out some of the questions that you had earlier in terms of transferring the neonatal, transferring of a compound. We can't figure out, what the lethal levels or lethal doses are. There's so much variability in it. So it becomes very difficult to answer a lot of these questions with a lot of certainty.
Representative Nelson.
Thank you. Wouldn't some domestic species kind of cross over? Like, if you talk to veterinarians and if a cat was exposed and the cat was lactating, do they transfer these properties over to their kittens? And, and my wife's a doctor and she says these anticoagulants to her are cakewalk to cure. Of course, she's working with domestic animals and vitamin K takes care of it. But somewhere there's got to be data on domestic animals that tells what levels are deemed too much. And someone has got to have done the studies. Do they pass it through in low levels? Or does it accumulate and accumulate? And, a 15-year-old Fisher, how old? I forget how old they get to be, but
Like maybe 10 to 12 years old.
Yeah.
That's a really lucky Fisher.
Yeah. yeah, well, we have a lot more cars on the road. But a 10-year-old Fisher, has it accumulated over time? Or, if they got it when they were younger, then, they got old and ran out of their prime territory and were out in the woods, do they pass it through? I mean, these are somewhere, someone's got the research on that, from domestic animals. And, I'm sure their inner workings are pretty similar.
I'm unfamiliar with any sort of data on domestic animals, unfortunately, and I'm sure we can look into it, of course. But yeah, I mean, in terms of time of exposure and how long it stays in their system, I think it depends on the type of compounds and how much they ingested and their overall health. So it's speaks to the complications of trying to understand this issue more, because different species have different metabolisms, right? A Fisher is going to have a relatively fast metabolism compared to something like a bobcat that is going to metabolize things a little bit differently, based on how much they're moving around, how much that they can find prey. Seasonality is going to affect their metabolism. So every species is going to be much different.
All right, I think we can keep going. Good question.
So, are there testing alternatives? Yes and no. So I had mentioned earlier that sure, you can take a blood sample and you can get amounts of, again, in parts per million of those different compounds, but it's not going to give you the full picture because the whole history of what they've been exposed to is going to be in their liver. So it would only give you kind of like more recent exposure. Hair samples and scat samples are very similar in that regard. The livers are essentially what bioaccumulation, right? That's where it all occurs. And the trapping information has given us that window to look into this. Otherwise, we wouldn't know the information that we know today. And as I mentioned earlier, we haven't noticed any sort of population level impacts. Even with this alarming information, we've looked into it upwards and sideways and all the different ways we possibly can, and we're not seeing any sort of population level impacts, which is really interesting. Other states, I don't believe, have seen any sort of population level impacts, and this is ongoing research. But it's important to note that ARs have been on the landscape for quite some time, and the population of Fisher we haven't noticed any sort of declines. So it lends itself to a lot of questions that are obviously very difficult to study. And just to complete this part of the presentation, so what we're doing now, so the Department of Fish and Wildlife, we're collaborating with other northeastern states as part of a broader effort to understand the health of gray fox and fisher populations. So as I mentioned earlier, there's gray fox population declines in a few different states, Vermont not being one of them. And so we're collecting carcasses, of course, and doing the necropsies, and testing for rodenticides is just one component of the study. We are also doing long-term monitoring. So we're going to continue to take liver samples from the carcasses that we receive from trappers, and we're going to continue to sample and send those samples off. And it would be particularly interesting, again, to look into trends at certain towns, compounds, and whether or not the exposure has increased or decreased. So it really gives us a very unique opportunity to look more into it. And we're also collaborating with the Agency of Agriculture, Food and Markets on developing a task force to figure out ways to minimize or reduce exposure. And so the information that we'll receive from the long-term monitoring that we have going on will help to advise in that task force.
Representative Nelson.
Yeah, are you checking for the bromophiolins? That's like in Tomcat, the one that goes to the central nervous system.
Non-anticoagulant.
The one that keeps my wife up at night.
I think it's bromephelin.
Bromephelin,
Yeah.
that's it. Thank you.
Yep. So at the time, no. Of that study, it didn't show up in any of the compounds that were tested. But we are going to be looking into that in the compounds, in the samples that we have now.
Because I believe those now can only be used by licensed pesticide applicators. So,
And bromephelin is far more toxic than any of the first and second generation.
it's the one that keeps my wife up at night.
The looks like the testing, the data that you shared with us goes back to 2018.
Mhm.
Is there was the state doing any research, any testing earlier than that?
No. As I mentioned earlier, we've had several decades of being able to collect these carcasses for necropsies to gather all of that health data. So we've contributed to a variety of different studies that are like contaminant studies, like mercury studies with otter, for example. And so this is just one part of the several different studies that we have going on to look into the health of these populations. So prior to 2018, we don't have any information, unfortunately.
And we, as John mentioned at the start of the presentation, we had heard some testimony last week on the impact on raptors, and recognizing that your specialty is fur bearers. Are there other fur bearers? Are we likely to hear that there are other species that are potentially impacted beyond the two that you mentioned here?
There's potential. Right, so the ones that we have results for are fisher and bobcat. Gray fox will have results for that soon, probably within the end of the year, hopefully by winter time. The other species that is interesting is otter. So I believe that I think that rodenticides have been detected in otter recently. So we have taken those samples and we've batched them along with our fisher and bobcat samples to test. So, but there's always potential for other species to be exposed as well, like coyotes and red foxes, for example. And if that were to come forward as a study objective, we would probably work with the trappers and their harvest. So to get that information.
There isn't any obligation for coyote trappers to turn in the carcasses.
No.
Senator Murphy.
But otter, isn't it primarily a fish eater?
Yes,
So there could be some in fish that some of this puts in.
I think how it's getting into the waterways, don't know. Of course.
Then they're vegetarian things like muskrats and beaver. Has anybody ever checked those to see if they have?
Not to my knowledge.
Everyone else?
No. Sad and interesting at the same time.
Aside from this, have you ever looked for PFAs? PFOAs?
As far as I know, I don't think so. I'm relatively new to this position. I've been in the position for about 2 years, but I think in the history of us having this program, I don't believe that we have.
Yeah, thank you.
All right, we may have exhausted our questions. No, good.
I'm very glad to hear that nobody said fish or cat.
Oh. Do you know where the term fisher cat came from?
No.
So when Europeans first came to the Northeast, they saw fisher, and there's something called the European polecat
Yeah.
that looks really similar to fisher, and so that's where the term fisher,
Switch to fish and cat.
even though they're not cats, they're mustelids.
Yeah, but they're big weasels.
They're big, feisty weasels.
And they live to be maybe 10 or 12 years old.
At best. Yep.
Are the carcasses that were sampled, do they span that entire range of years or?
I think so. I have to double check. I think the majority of our harvest tends to be juveniles and juvenile males in particular, because they're moving across the landscape more, and they're just trying to figure out how to get from one place to another and find resources. So they are typically more vulnerable to traps than adults are. I would have to check on the age distribution, but generally speaking, every single year, our age distribution is typically younger individuals that are being harvested.
Just like juvenile and males, they just aren't smart enough.
There you go.
All right, well, was there anything else that you wanted to say?
I would just say that I hope that from this presentation, I hope that you all can see that we're committed to this issue and dedicated and trying to figure this out more. It's not an easy thing. Like I said, I mean, we don't have the opportunity to have laboratory conditions. So we're constantly working on trying to figure out different strategies at getting at some of these answers more. And it's going to take some time. It's not going to happen overnight.
I think, I think the question you asked, Representative Nelson, is interesting, whether there's data out there on other lactating species and mammals, whether that might be informative. Representative Lipsky.
I have one more way outside the box question, but I recently visited a major zoo the last 5 months in Prague, and they're very natural habitats. Do you know of any zoos that might have fisher that have ever tested a deceased fisher for liver content anywhere in the US?
I do not know.
So a fisher that wasn't raised in the wild.
Or they may have spent some time, but the habitats at the zoo in Prague were extraordinary, and they're in captivity, and if they die, they're possible to be tested. That's
You were
all.
in Prague?
For Veterans Day. That's another, that's, you don't need it. It's just, but I had grandkids and I took them to the zoo.
All right, well, we can save that story for after we release our witness, but
Just one last thing too, it's that as we continue to study this, we're happy to update the committee as well. We're not gonna have results right away, but we're getting ready to send out a batch of samples soon. And when we do that, we should have results, like I said, by the end of the year. So, and again, we're working on a broader study, so if you would like a legislative progress report or would like for us to come in again and testify, we're happy to do so.
but yeah, I think that would be interesting. I don't know that we need to put it into a bill, but if you're offering it, it'd be, yeah, certainly take you up on that.
So as far as you know, at this time, scientists or whoever does that are working on any alternatives that aren't as destructive as this. Do you know that if studies are going on in that respect?
I don't, like alternatives to pesticides?
Yeah.
Yeah, I'm not
That are
certain. about-
pesticides, but not ones that do what these do.
Yeah, I'm not certain on that. That could be a topic of discussion for our task force to look into, but I don't know.
Thank you.
Great, yeah. Thank you very much for coming in.
Yes, thank you.
Thank you.
Thanks for having us.